I would like to introduce Scott Coffin, who is our featured keynote speaker this morning. It's really my pleasure to introduce Dr. Coffin. He holds a PhD in environmental toxicology and serves as a research scientist at the California State Water Resources Control Board. Since 2014, Dr. Coffin's research has focused on microplastics toxicity and characterization. Currently, Dr. Coffin is leading California's development of drinking water regulations for microplastics and advising management strategies in aquatic ecosystems. He's recently led international expert groups to assess risks of microplastics to humans and aquatic ecosystems, and to harmonize analytical methods for microplastics. So today, Dr. Coffin is going to speak to us about his work on the risk of microplastics to both ecosystems and drinking water. So feel free to add your questions for Dr. Coffin to the Zoom Q&A. And with that, please join me in giving him a warm virtual welcome. And I will stop sharing so that you can start sharing.
Thank you for that nice introduction, Sarah.
You're welcome.
Really happy to be here today.
The floor is yours.
Thank you. Alrighty, so we'll get right into it. So today I'll be talking about some of the research that we've been doing here in California to assess and manage risks of microplastics in ecosystems and in drinking water. The plastic waste crisis has recently gained a lot of attention in the media. I'm sure these images look familiar to you by now. Everywhere scientists have looked, we have found microplastics, all the way from the deepest part of the ocean to the tallest mountain in the world. This increasing pollution has caused public awareness of the issue to increase as well. This is best illustrated by this comparison of a 1997 advertisement from the American Chemistry Council stating that plastics are an important part of your healthy diet, in contrast with a 2020 cover of Consumer Reports saying how to eat less plastic. Excuse me. As plastic enters the environment, it doesn't just go away. In fact, it breaks down into smaller and smaller particles down well into the nano-size range. You can actually visualize the breaking off of nano-sized particles from this facial scrub particle shown here, looking at the 1 micron scale. If you look very closely, you can see these nanoscale particles breaking off. So this increased awareness and concern for microplastic pollution, for the environment, has caused the California legislature to pass 2 important bills in 2018. Senate Bill 1263 requires this State Water Resources Control Board, where I work, to work with the Ocean Protection Council to initiate a statewide microplastic strategy by 2022. By 2026, we are required to develop a risk assessment framework, get standardized methods for monitoring, establish some baseline occurrence data of how much microplastics are in our marine environment, investigate the sources and pathways into the environment, and then go back to the legislature with some recommended source reduction strategies. In 2017, the first study published on microplastics in tap water found particles in 94% of samples in the United States and 83% of samples worldwide. This caused quite a stir and prompted the California legislature to pass Senate Bill 1422. This is very similar in nature to Senate Bill 1263, the difference being that it's focused specifically on drinking water and it has a shorter timeline. By July 1st of 2020, we were required to define microplastics. And by July 1st of 2021, we were required to adopt a standardized method for monitoring, a plan for 4 years of testing and analysis, consider adopting a health-based guidance level so that when we tell consumers how many microplastics are in their drinking water, they know how to interpret that information, and provide accreditation for laboratories. So defining microplastics is not a trivial task. This is one of the most complex, if not the most complex, contaminant suite scientists have encountered. It's made up of billions of different combinations of particles with different chemical compositions, shapes, sizes, colors, and associated contaminants. Despite the challenges of writing a definition that encompasses this very broad class of particles, we accomplished this based on some harmonization that has happened in the academic literature. And in 2020, California passed the world's first regulatory definition of microplastics, which are solid polymeric materials to which chemical additives or other substances may have been added, which are particles which have at least 3 dimensions that are greater than 1 nanometer and less than 5,000 micrometers. Note the very large size range here. Polymers that are derived in nature that have not been chemically modified other than by hydrolysis are excluded. So that is to say, natural particles are not considered microplastics. So getting right into the health effects research that we conducted to address the legislative requirements of Senate Bills 1263 and 1422, we conducted a year-long workshop with experts from around the world on microplastics and toxicology to conduct a meta-analysis of the available toxicity information. Our main questions were to determine if there are certain particle characteristics that influence toxicity, determine what the adverse effects of microplastics are in both aquatic organisms and in humans, and develop a threshold framework so that we can interpret this varying levels of information in a management context. I'll talk first about the ecological risk assessment that we performed, again, to meet the requirements of Senate Bill 1263. Our expert work group identified 2 ways in which microplastics can cause harm. The first is chemical hazards, and this is really the plastic associated chemicals. We know a lot about some of these. Bisphenol A and all of the analogs, PFAS, brominated flame retardants. A lot of these chemicals have actually been studied for several years, if not decades. And we have a fairly good understanding about their hazard traits as well as how they flow through ecosystems and through the environment. The other mechanism is through the particles themselves, which much like PM 2.5 or PM 10, other very small particles, they can cause harm through well-known mechanisms, but there may be some other mechanisms in which they can also cause harm. A problem that we identified in relating the laboratory studies to what we find in the environment is that in the environment, we have a polydispersed mixture of plastic particles, extremely broad, with what you could describe as a fruit basket. There's many different shapes and sizes and colors, much like many different types of fruit in a basket. In the lab, they're typically using just one type of fruit. And what I mean by this is they might be using a polystyrene sphere of exactly one size or polyethylene. So we have a lot of information about exactly that type of fruit, but we don't understand exactly how all of the fruits together cause harm to organisms out in the real world. There's been some recent advances in the literature that have allowed us to overcome this non-alignment problem. In 2019, Cooey and Coleman's published a paper that uses probability distributions based on the size, density, and the shape of the particles to mathematically solve some of these issues between relating the polydispersed and the monodispersed mixtures. Critical to making these relations is understanding how the particles actually cause harm. And this is well predicted by the particle traits. We know that microplastics can cause what's called food dilution. Basically, the particles take up space in the gut and they prevent the organism from getting energy from the other food that they consume. This is well predicted by the volume of the particles. And we restrict the bioavailability of the particles based on what they can actually eat, the size of their mouth. The other effect mechanism is a little bit more well known with particles. This happens through tissue translocation of the particles. And this is really restricted to the very smallest of sizes, those that can actually translocate across the tissue and accumulate in our organs. And this is well predicted by the surface area of those particles. Now that we are able to relate the polydispersed and the monodispersed mixtures of microplastics, we can use traditional ecotoxicological risk assessment tools to predict the ecosystem level effects. And to do this, we used a species sensitivity distribution approach in which the least sensitive and the most sensitive species are plotted on a cumulative distribution plot, and we choose a percentile of this model to set the risk thresholds, typically the 5th or the 10th percentile. We derived a range of ecotoxicological thresholds for microplastics based on the certainty of hazard or the certainty of risk occurring in the environment. This goes from a protective to a predictive a model, with the lower tiers corresponding to inexpensive management options, like just doing a little bit more monitoring. The upper tiers are tied to more costly management actions, as we feel more certain that the effects are truly occurring in the environment. And we derive these thresholds both for food dilution effect mechanism as well as for tissue translocation. We realize that different government organizations are going to have different ways that they want to derive these thresholds. And this depends on the specific species and their ecosystem, as well as the regulations that they may already have in place for deriving thresholds using SSDs. The mathematics and coding required to derive these thresholds are not trivial. And we wanted to make sure that the coding aspect would not be a barrier for anyone to develop their own thresholds. So we developed an open source, open data platform called TOMEX, or the Toxicity of Microplastics Explorer, that includes the ability to derive one's own species sensitivity distributions using the microplastics toxicity database. What I'm showing here is just a brief walkthrough. You can see that in less than 1 minute, you can derive your own species sensitivity distribution and use this for the ecosystem of interest. This application will be publicly available within the next week. Actually, we just published the publication underlying this, and this is also a living repository. So when studies get published, they can upload the toxicity data directly to the application, and so this application will be continuously updated. So now that we have thresholds, we can actually apply them to monitoring data in the environment to assess risk. We recently completed a risk assessment for San Francisco Bay. There's been some monitoring done there in which they used Mantitrol to identify the microplastics at different locations. And when we apply these thresholds, we find significant risk within the central part of the bay, with limited risks outside of the bay. This indicates an accumulation of microplastics due to the low flow and the low dilution within the bay. No assessment goes without uncertainty, and it's important to present these uncertainties in the context of management in particular. So we calculated the uncertainty of this risk assessment probabilistically using Monte Carlo modeling, and determined that a significant number of locations in the San Francisco Bay exceed this first threshold. For most samples, the uncertainties were simply too high to conclude that the additional thresholds were exceeded. So this suggests that the bay deserves additional monitoring. However, it might not be ready for more heavy-handed management options. A similar risk assessment was recently completed on the global scale and extrapolated to the year 2100, assuming we continue with a business as usual production of plastic. The study found that currently only 0.17% of the world's oceans have already exceeded risks for microplastics. However, this increases to 1.62% of the whole ocean by 2100, with enclosed water bodies like the Mediterranean and Yellow Sea expected to have much higher risks than out in the open ocean. So now that we're armed with a quantitative framework to assess the risks of microplastics, we can develop a long-term strategy to assess and manage risks in the ocean. To manage risks of microplastics in the marine environment, the Ocean Protection Council adopted a strategy in 2022 that takes a two-pronged approach, which includes some multi-benefit, no-regret solutions to act on the problem now, and science to inform future actions. The strategy is quite bold and ambitious and will require input from many stakeholders in all sectors over the next few years. The strategy science needs call for monitoring in the environment to assess risks, identify sources and pathways, and then develop solutions based on those findings. This is an iterative process, and by repeating this on a continual basis, we can monitor the success of these intervention strategies and improve them. The statewide strategy specifically identified microfibers shed from clothing to be a significant source of microplastics in the environment. A recent study estimated that 2.2 kilotons of synthetic microfibers were generated by apparel washing in California and displaced into the environment. 74% of these microfibers entered the terrestrial environment through the application of biosolids, with only 5% going out into the waterways. When plants are exposed to microplastics in soil, we know that they grow more slowly and they can produce less fruit. So this may be cause of concern for food security as well as our terrestrial ecosystems. The authors of that 2020 study also determined that these impacts are borne primarily by low-income communities, which receive the bulk of contaminated biosolids that are generated by high-income communities in California. You can see on the left is where the biosolids are generated, and on the right is where the biosolids are sent, typically to the Central Valley, which is much lower income. Solutions exist already to reduce the microfiber loadings into the wastewater treatment plants so that they don't make their way into biosolids or out into the waterways. Installing washing machine filters in 10% of homes in a small town resulted in a significant reduction of microfibers in the wastewater treatment plant effluent. Currently, the California legislature is considering a bill that would require all new washing machines to come pre-installed with these filters. So I've talked a bit about the aquatic ecosystem, and I'm going to shift gears and talk about microplastics in drinking water and the human health risk assessment that we conducted. Recently, microplastics have been detected in lungs, blood, and human placenta, confirming that we are being exposed and that the particles are not simply being egested right away. They are actually accumulating in our organs. And the billion-dollar question here is, what does this actually mean for our health? Luckily for us, there's been an exponential increase in the number of toxicity studies performed on mammals, starting in 2018. When we began our analysis, we were able to scrape this literature and conduct a comprehensive meta-analysis. We found 29 ingestion-based in vivo mammalian microplastics toxicity studies. All of these used rodents and exposed them to a single size, shape, and polymer of microplastics. Most of these were done using polystyrene spheres. We then screened these studies through a quality assessment screening and prioritization model using experimental design, particle characterization, and risk assessment applicability traits. And we were left with 12 studies that we deemed fit for purpose for further review. We then subjected these 12 studies to additional scrutiny using experts in the fields of these particular studies. Of these reliable studies, only 4 reported effects on male reproduction, 2 reported effects on female reproduction, and 1 study reported effects on the liver. These studies consistently showed adverse impacts to sperm deformity and count, as well as decreased testosterone in male rodents. We then used this information to derive dose response modeling using EPA's benchmark dose modeling software. The point of departures for each critical effect are shown here, with colors corresponding to the different studies. The most sensitive effects were for testes weight and anti-Mullerian hormone concentration, which is a biomarker related to female reproductive impacts. We also observed a similar range of concentrations in which other effects occur, especially male reproductive impacts such as sperm maturity and viability. Despite these findings of consistent impacts to male reproduction in rodents, there's some significant data gaps that currently prevent the reliable assessment of microplastics risks to humans. Primarily, there are 3 classes of problems, including an inadequate effects database. I mentioned before, most of these studies were using polystyrene spheres of a single shape and size, and they typically did not characterize the particles very well. Additionally, there is evidence for oxidative stress and inflammation being linked to the toxicity that we observed in the rodents. However, uncertainties remain about how these effect mechanisms are truly occurring. Once we can understand the effect mechanisms, we can extrapolate these to the diverse mixtures that humans are being exposed to. Lastly, an incomplete exposure database represents an important data gap with limited information for microplastics in our diets and no harmonized drinking water data. The State Water Board is currently monitoring and supporting additional science that is needed to assess the human health risks of microplastics. In particular, we need exposure data on what we are drinking, breathing, and eating, and we need high-quality laboratory toxicity studies to determine how the effects are occurring and at what concentrations they occur. So now that we've covered the health research, I'll talk a little bit about the state's efforts to go out and monitor microplastics. And to do this, we first need a standardized method and be able to accredit laboratories for using these methods. Before Senate Bill 1422 was passed, there were no harmonized methods for monitoring microplastics. To address this gap, we worked with the Southern California Coastal Water Research Project to develop standardized methods using two types of instruments applicable to drinking water, ocean water, fish tissue, and sediment. The method performance was evaluated through an interlaboratory validation study involving 26 laboratories from 7 countries. The laboratory process for analyzing microplastics is relatively straightforward. Microplastic particles are first extracted from a sample using filters, and particles are identified and categorized using a microscope. The pictures and measurements of microplastics may be taken for further analysis, but in order to determine if the particles are plastic or not, it's critical to confirm using a chemical analysis technique. The common techniques include light-based spectroscopic techniques such as Raman or infrared, as well as pyrolysis, gas chromatography, and mass spectrometry. The laboratories were sent blind samples that contain realistic mixtures of particles found in drinking water. 4 common plastic polymers were used, each in varying amounts in 4 size fractions and in 4 different shapes, ranging in sizes from 1 to 1,000 microns. Additionally, false positive materials like sand and shell fragments were added to test the robustness of the identification methods. As proficiency testing samples for microplastics were not commercially available at the time, we worked with an organization in Norway to specifically develop these samples for this project. So let's first look at the overall findings of this study. On the y-axis, you can see the percentage recovery, with a dotted line showing where the 100% mark is. The closer we are to 100%, the more accurate the measurements. And you can see that the grouping of the data reflects the precision as well. Looking at the pane on the left for the total particle recovery, there was relatively low recovery and low precision, but this is not the whole answer. Let's look at the second panel from the left, which breaks the data up by the size fraction. Here we can see that precision and accuracy are good for particles that are larger than 20 microns. In the third panel, we see higher recovery than expected for blue and clear particles, and lower than expected recovery for orange and white particles. A similar trend is seen with shapes on the final panel, with higher than expected recovery for fibers and lower recovery for fragments. Looking at the overall strengths and limitations of the assessed methods, we feel that we have 2 methods that are ready to use for drinking water analysis, including infrared and Raman spectroscopy. IR and Raman are highly accurate for identifying plastics, while optical microscopy has limited overall accuracy. The average measurement time per sample is higher for Raman than infrared at 15 hours compared to 10 on average. Additionally, infrared instruments are generally cheaper than Raman. Consumables can also be more expensive for Raman on average. The main strength of Raman is that it can identify smaller particles with an approximate lower size limit of about 2 microns. So we recognize that spectroscopic methods are slow and expensive, but they provide high resolution information. To reduce the cost for monitoring, we are developing a tiered monitoring framework that would use a surrogate screening method to determine if additional monitoring is warranted. A potential second-tier method would involve a mass-based determination, and if necessary, samples would undergo complete characterization using Raman or FTIR. The standardized methods that we developed based on Raman and infrared spectroscopy can currently be found on the State Water Board webpage. In addition to having a standardized method, laboratory accreditation is key to obtaining reliable monitoring data. California's Environmental Laboratory Accreditation Program, or ELAP, will be providing accreditation to qualified laboratories across the US starting this summer. So now that we have defined microplastics, developed a standardized method, assessed the health effects, and can accredit qualified laboratories, it's time to go out and do some testing in drinking water. In designing a sampling program, it's important to know how the particles are moving through the treatment systems. Treatment removes microplastics based on the filter size. Most drinking water treatment plants that have surface water for their sources use filtration methods to remove particles, and they may incidentally be removing microplastics already. Flocculation and sedimentation can remove particles as small as 1 micron, which is about the size of a red blood cell. More advanced filtration methods, such as microfiltration and reverse osmosis, can remove even smaller particles. As I showed earlier from our method validation study, we can reliably quantify microplastics as small as 20 microns. So we can't currently monitor microplastics that are small enough to make it through most treatment techniques. We're working to improve these methods, and in the meantime, our current methods can be used to characterize the contamination in the source waters, which will be an instrumental first step in determining potential exposure to humans. We are developing a phased monitoring approach for monitoring on microplastics. The first year would be a pilot phase paid for by the state, which involves a sample method comparison project, range finding of contamination, and sample training for system operators. Following the pilot phase, we would issue monitoring orders to a limited number of systems to document contamination in their source waters only. As part of this phase, we would develop a surrogate method if one is available. A final phase would explore the additional sources and look in treated waters using the tiered monitoring approach. In this phase, we would also monitor in the treated effluent that's actually entering the homes. So the complexity of microplastics characteristics allows for really complicated and useful large data sets with each particle characterized in the actual data set. However, there's no harmonized data reporting, which makes it challenging to upload this data in a way that will be useful on the back end. We're partnering with the Moore Institute for Plastic Pollution Research to develop an open source data intake platform for microplastics that would harmonize the data reporting and integrate with existing data repositories the state already manages. This is a draft schematic of this data processing pipeline. The project will be completed within 1 year, and because this code is going to be open source, anyone around the world will be able to use and adapt it to their own needs. So if you're conducting research on microplastics or would just like to learn more about what California is doing, I invite you to join our working group. I lead the Microplastics Monitoring subcommittee of the California Water Quality Monitoring Council, which is a local and global community exchange of information and data for microplastics monitoring methods and tools. We're currently developing a sampling and analysis playbook, a risk communication toolbox, and a laboratory accreditation and data analysis assistance program. So this is, we meet quarterly and anyone is able to join. If you're interested in getting involved, please shoot me a message. So while California is making great strides to address plastic pollution, we recognize that this is a global scale issue that requires coordinated international efforts to truly address. Currently, there is a patchwork of policies and regulations to address plastic pollution. On our current trajectory, we will double the amount of plastic we create by 2030. International binding treaties on plastic, such as MARPOL in 1973, which banned the practice of ships dumping plastic into the ocean, have significantly reduced the plastic that we're finding out in the environment. However, the last legally binding international treaty for plastics was reached in 1991 with the UN Fish Stocks Agreement. All of the subsequent agreements have been voluntary, and therefore they've been minimally effective. The adage that an ounce of prevention is worth a pound of cure holds true in the case of plastic pollution. By focusing on upstream mitigation efforts, such as bans, we can avoid the costly and ineffective mitigation techniques of trying to clean up plastic once it's already entered the environment. It seems the world leaders have been listening to the scientists lately, and this year at the United Nations, they recently agreed to negotiate on a global treaty on plastics that would be binding and would address the increased production that we're predicted to have. This progress in the international policy world gives me hope as a scientist that the work that I do can make meaningful change. So thank you all for your attention, and I look forward to chatting with you here as well as over Twitter.
Thank you very much. That was really excellent. As usual, California is helping to lead the way on this clearly. Janice, I will turn it over to you to guide our questions, because we definitely have time to take some questions. So please, everyone, feel free to put your questions for Dr. Coffin in the Q&A and upvote the ones you think are the best, and we'll make sure that we get to them. Janice?
Fabulous, thank you. Our first question is, why are many federal government organizations not talking about these risks? Oh, my screen, sorry. None of their education materials or research they fund address these human health concerns. They always seem to avoid this topic.
So I can only really opine on that because I don't work for a federal organization. I don't know what kind of conversations they're having internally. But what I can say is that a lot of the research for human health effects of microplastics has only been published within the past 2 years. It took us about a year to do a comprehensive meta-analysis, and I can only imagine that the federal organizations that didn't already have experts within house to assess these studies are just trying to keep up with the rapid pace of the literature. That being said, there has been a very small amount of funding from the US federal government for the human health effects of microplastics. During our review, we searched federal databases for grants that have been issued for this particular topic, and we found only one, and it's a pilot project worth about $100,000. The European Union has already dedicated $30 million to assessing the human health effects of microplastics through grants that will be completed by 2024. So we're really looking forward to the research happening that'll be happening in Europe. And I believe that we will have a good understanding about the human health risks of microplastics within the next few years.
Fabulous, thank you. Our next question is, are there any ongoing studies that delve into the long-term leachability of the chemical components of microplastics at different environments? For example, ocean, treatment plants, sewers, etc.?
There's been actually quite a few studies on the leachability of the chemical components. In 1970, the first finding of microplastics in the ocean was actually in the Sargasso Sea. The principal concern in that study was the leaching of chemicals from the plastics. They also found PCBs and DDTs adsorbed to these plastic spheres. So scientists have been thinking about the chemical risks of plastics from the very beginning. And actually some of the first studies that were conducted on microplastics in laboratories focused primarily on these leachates. So we have actually a pretty good idea about the dynamics, the chemical exchange dynamics, the fugacity of these chemicals between the particles. And it seems that in most cases, the chemicals that are sorbing onto plastics, like the PCBs that are already out in the environment, the plastics are not serving as a relevant vector. So once an organism ingests that particle that has PCBs, it's not relevant compared to what they're getting from their food, for example, which is just as contaminated. The leaching of the additives like BPA and PFAS, for instance, that may be a more relevant exposure route. And there's a number of ongoing studies in the US and elsewhere that is trying to address the relevance of that pathway.
Excuse me. And as a follow-up, the same question asker was curious if the physical removal of these particles might mitigate these chemical risks.
It's possible. So one of the things that we found is that when the plastic particles go out into the environment, they tend to rapidly leach out the chemicals that are attached to them, or sorry, that are added to them. And so within the first month or so, if they're getting hit, especially by UV light as salt, like they are in the ocean, they're going to leach out the majority of those chemicals. So going out and collecting the plastic after it's already been sitting out in the open ocean, for instance, for a long time, you're probably not going to get a whole lot of those chemicals. They're probably already leached out.
Okay, and similar vein, but moving to fibers. Once the fibers are removed by the filters from textile washing, then what is done with the material? Are we not just releasing it back to the environment?
So the proper handling of that material is critical to this solution. And it's important that the fibers get captured into a container that can no longer break open. And really the only place that we can send those right now is the landfill. The landfill is a better place than the open environment and our drinking water, but it's obviously not a very ideal solution.
Excellent. Next question is, do you accredit labs outside California for these methods?
Absolutely, we can accredit laboratories anywhere in the world. We recently trained third-party assessors that are not working for the state of California, for private companies, to accredit laboratories for our methods. And so while the state of California is currently the only government agency that oversees accreditation, independent assessors currently have the ability to go and accredit any laboratory for these methods.
Interesting. Our next question is, what do you think about the current research interests trending from microplastic towards nanoplastic? Can we expect relevant facilities to respond in making standards and policies for nanoplastics soon?
So it's been really challenging to identify nanoplastics in the environment, which is the key question right now. We have a ton of laboratory toxicity studies on nanoplastics, and it was actually about a third of the entire database that we had for both human and aquatic eco risk. We can't do anything with those studies right now because we have no idea what we're being actually exposed to in the environment. So really this comes down to an analytical challenge. And once we are able to adequately monitor for nanoplastics in the environment, the field will rapidly get up to pace in terms of assessing risks and developing standards. And I anticipate that'll be within the next few years.
Thanks. All right. We did have a question about if you are able to estimate concentrations from standardized methods. How long does it take you to identify each type of plastic from each particle?
So the the standardized methods that we have using spectroscopy are particle counting methods. And using these methods, we can estimate the concentrations within a given volume. We know the volume that we filter in order to get those particles. And we also know we can also identify the polymer, and that tells us the density. So if we know the particle shape, size, and density, we can estimate the mass as well. And so we can now interchange between mass, volume, surface area, particle count, even specific surface area, which is a good predictor of inflammation, with just the one method using spectroscopy.
Awesome. Now, another question chiming in about remediation. How can we get rid of existing microplastic pollutions downstream as the numbers and effects accumulate?
So the further you are to the source or of the pollution, the easier it's going to be. There's a number of techniques that can remove microplastics from rivers. The ocean cleanup is working on that. Stormwater is a highly relevant pathway of microplastics into the oceans, and we can address that with green infrastructure, low impact design. These types of infrastructure are already in place to remove other chemical contaminants, and they work really well for microplastics as well. Once they're already out in the environment, cleaning them up may be more, it may cause more harm than good. What we're seeing with the ocean cleanup, going out with these big boons in the middle of the open ocean, is that they're trapping wildlife in those nets, which may be causing more harm, and also they can create additional debris when these massive systems fail in highly complex systems out in the open ocean. So I think focusing as close to upstream as possible is going to be the best route.
Wow, fascinating. Can you comment on the difference in removal percentage based on the color of microplastics?
Yeah, so for the standardized or the interlaboratory validation study, some of these laboratories had contamination from their clothing. And that is particularly the fibers shedding from the operator's clothing is what was causing the higher than expected recovery for the fibers. The colors also had to do with the lab coats they were wearing. So some of them were wearing blue, like flame retardant, or sorry, the fireproof lab coats. And that's why we're seeing the higher recovery rates. Our laboratory accreditors are paying very close attention to the controls within the laboratory to reduce background contamination. And we have within our standardized method a number of blanks that are required by the operator, the laboratory analyst to run, including an air blank. Anytime they're doing any sample analysis or processing, they have to have an air blank open. So that collects all of the fibers from their clothing and from the carpet, if you have any. Ideally, you don't have anything in your lab that's made of plastic. All of your furniture should be metal or wood.
Wow, it really kind of reminds you how much plastic is everywhere. We had one more question, and I'm probably and not as familiar in this area, but PY-GMMS is commonly used for analysis of microplastic. Is that a particular procedure you're probably more familiar with than me? What are your thoughts on the role of PY-GCMS in the analysis of nanoplastics?
So PY-GCMS is pyrolysis coupled with gas chromatography mass spectrometry. And GCMS is one of the most commonly used instruments for detecting dissolved chemical contaminants. A lot of analytical laboratories are running these for PCBs and other things that we've been monitoring for decades. And so the advantage is that we can buy a relatively inexpensive device called a pyrolyzer, hook it up to the existing GCMS, and now you're able to analyze the mass of microplastics and nanoplastics. This is a really exciting development, and I believe that it will be useful for analyzing not only microplastics, but also nanoplastics. We had 2 laboratories that performed a standard protocol that we developed for pyrolysis GCMS in our laboratory study. But because we only had 2, we couldn't do any statistics. We really need a minimum of about 7 laboratories to feel confident about the results. So we hope to repeat this study in the next few years with more laboratories running by GCMS, and hopefully we'll be able to use that method.
That wraps up the questions in my question and answer box. Thank you for your rapid fire.
Yeah, that was really great. We really grilled you, I think, on that one. I have just sort of a question, and maybe it can be the final one. We're almost out of time here, but I'm just sort of thinking, if taking all of this into account, I mean, what happens next, right? We've established that these risks exist. We know that we're going to be seeing more research come out in the next couple of years about the impacts to humans. We know that there are ecosystem impacts. So then what? I mean, can you just kind of, and I don't expect you to have any answers, obviously, but can you just kind of reflect a little bit on what you think the future of this looks like?
So one of the reasons that government agencies have not adequately addressed the problem of plastic pollution is because we haven't been able to fit it in our existing regulatory frameworks. And really, it's like a square peg in a round hole when you think about it, because it's not one thing and it's everywhere. Everyone has it all the time. It's almost like PFAS or like lead. And you can't point the finger at any in particular entity. It seems like it's a societal thing. It's actually a fundamental building block of modern society. So that's going to cause us to dramatically rethink how we use this material and use any material, whatever we substitute plastic with, whether it be a bio-based polymer or just a natural particle. We're going to have to evaluate if that is more or less sustainable than what we're already using. And so this is going to require a lot of science, as well as a lot of informed policy to address this issue.
Yeah, it's interesting. It is incredibly complex, and we've really only scratched the surface of it. And it'll be interesting to see where we're at in 10, 15 years, and with how ubiquitous plastic is now, and is it going to continue to be? So, all right, well, we are right at time. I thank you so much for your time. That talk was excellent, and I appreciate your ability to field those rapid fire questions, because Janice really hit you with some. And thank you to all attendees for your thoughtful questions as well.